Hello everyone, I am Srija Chakraborty, a student of the Indian Institute of Technology, Delhi, and this is my presentation for our project titled Identification and Imaging of Potential Vaccine Targets for the SARS-CoV-2 Spike Protein using the Machine Learning and Bioinformatics tools. This was a collaboration between me, Tharinda Karawita, and Thavisha Herath, and was completed under the mentorship of Dr. Mohit Mazumder and Dr. Elia Brodsky of the Biotech Laboratories. For the past 2 years, the world is facing an onslaught of the COVID-19 outbreak caused by the novel coronavirus, the SARS-CoV-2. There is an urgent need for immunological study to understand this new virus, and to develop ways to control its spread. And our aim in this project is to gain insights for the vaccine design against SARS-CoV-2 by considering the genetic similarities between SARS-CoV-2, SARS-CoV, and MERS-CoV. And these targeted antigen sites have potential benefit for protection against the novel virus. Now, like SARS-CoV and MERS-CoV, the recent SARS-CoV-2 belongs to the Betacoronavirus genus. Hence, we know that studying the SARS-CoV and MERS-CoV could be a milestone for SARS-CoV-2 immunological study. Now, in our study, we have focused particularly on the epitopes in the structural protein due to their dominant and long-lasting immune response that was previously reported against the SARS-CoV. This is a detailed overview of the procedures that we have followed by us. Now, let us look at these steps in a little bit more detail. Now, firstly, we conduct a phylogenetic analysis of the SARS-CoV, MERS-CoV, and SARS-CoV-2 to find genomic similarities and differences. We run a quasi-species virus pipeline with the FASTA files of the SARS-CoV, MERS-CoV, and SARS-CoV-2 amino acid sequences of the spike protein, which we obtained from the NCPI protein dataset. And here we have shared a screenshot of the pipeline that we ran. Next is the acquisition of the epitope data. This we obtained from the NAAID virus pathogen analysis and database analysis resource, that is VIPER. We searched for immune epitopes under the coronavirus taxon, and the results were filtered to give experimentally determined epitopes of the spike glycoprotein betacoronavirus for the human host, with at least one positive assay each for the B cell, T cell, and MHC type assays. Now, the filters resulted in 518 epitopes, and we downloaded them in the form of an Excel file. Here, the first table shows the filtering criteria corresponding to the MERS-CoV derived epitopes from the VIPER, and the second table shows the filtering criteria, the corresponding number of SARS-CoV derived epitopes that again obtained from the VIPER database. This step is known as the epitope conservancy analysis. Here, the Excel files of that we obtained of the epitopes have to be reconstructed. They have to be filtered for different types of assays and converted into a FASTA file, and then uploaded to this IEDB conservancy analysis resource, which is shown here. The epitopes were then aligned with the SARS-CoV-2 spike protein reference amino acid. Now, this step is the epitope clustering. Here, the output that we obtained in the previous step was filtered to obtain epitope sequences with minimum sequence identity threshold to be 70%. And this was performed using the IEDB epitope cluster analysis resource, which is a tool that groups the clusters based on sequence identity. Here it was 70%, as I mentioned. And lastly, we imaged the sequences of the epitopes onto the SARS-CoV spike glycoprotein, and all the epitopes are matched into the 3 separate SARS-CoV spike glycoprotein modules. And finally, our results, and this is a brief overview of the results that we obtained, and we'll discuss these results in detail. The first result is that the spike glycoproteins of SARS-CoV-2 is genetically similar to the SARS-CoV, but not to the MERS-CoV, based on full-length genome phylogenetic analysis performed in relation to the spike glycoprotein. And this figure here shows this phylogenetic analysis that confirms the deviation of the MERS-CoV from the SARS-CoV and SARS-CoV-2. In the epitope analysis earlier, we showed that there's very low genetic similarity between SARS-CoV and MERS-CoV. On the other hand, when we replace the MERS-CoV with SARS-CoV, we find that we classified them into 2 different assays, the T cell assays and the positive MHC binding assays. We obtained about 362 T cell assays out of the 61 epitope sequences were identical for SARS-CoV-2, which have been shown in this table given here. And this shows the 33 epitopes of identical epitopes of the B cell assays. And next, we have the epitope clustering result. When we performed the T cell epitope clustering, 61 epitopes that we obtained earlier were diverted into 25 different cluster groups. And when B cell epitope clustering was performed, the 33 identical epitopes earlier were diverted into 22 different cluster groups. And we can see this result in the table here. This table here shows the T cell epitopes, and this shows a cluster of the B cell epitopes. Finally, we mapped these clustered as well as the singleton epitopes into one spike glycoprotein from the protein database, PDB, and 6DXX was the spike glycoprotein that was chosen for mapping. And when we mapped all these epitopes, we found that some of the epitopes are actually located quite close to the receptor binding domain. And this table here, it shows the SARS-CoV derived B cell and MHC binding epitopes that were located close to the RBD, and which were total 4 epitopes. Now, this shows the SARS-CoV derived MHC binding epitopes mapped onto the spike glycoprotein crystal, and here these are 3 views, the front, side, and top. As we can see, the blue regions are the epitopes located near the RBD, and the green regions are located inside the spike glycoprotein. This is for the T cell epitopes mapped onto the spike glycoprotein crystal. Again, the green regions here show all those which are present inside the spike glycoprotein, so there's nothing close to the RBD, and we have 3 different views: side, top, and front view. And finally, these are the B cell derived epitopes that are mapped onto the crystal model, and again there are 3 views: front, side, and top. The red region shows the epitope that is inside, and the blue region shows those near the RBD. These are the references that we consulted. Our major source of inspiration was the preliminary identification and potential vaccine targets of the COVID-19 coronavirus SARS-CoV-2 based on SARS-CoV immunological studies conducted by Ahmed, Kadir, and Mackay. Thank you very much.